Um, like today, we'll probably end class at 10, 15, maybe 20 minutes, I don't know, early. Me and Abby have a funeral to go to at 1, and it's 2 hours away, so we'll be leaving, like, we'll say 10:35. But I will get you all something tonight. However, you all have all the material in your lab manuals. But the way that I see study guides as is that you all should be studying with your lab manuals or whatever material you have, and then when I give you a study guide, it should be more like a checklist. Okay, I know these things, I've already studied all of them. And so, hopefully it'll be good for you all tonight. Just study what's in your lab notebooks. And of course, most importantly, like the organ systems, you need to, be able to look at those organ systems and be able to identify what it is. Maybe identify, similar to a quiz, like identify an organ or two from each organ system. I would probably recommend knowing all of them because you don't know which ones could pop up. And I'll look. I think I have the questions on my computer, and I'll make something for you all. Does anybody have any questions from last lecture, or of course, if you have any questions about the lab? Does anybody? Okay. So we left off talking about DNA, and just a little bit of a review. We'll look at this structure again. However, very important to understand is that DNA is a double helix, okay? And connecting these double helices together are hydrogen bonds. So each one of these portions, again, you need to be thinking in a mind of, and this is an artist's depiction of what's going on. So what's here is this structure here. Okay. Now the difference in G, C, A, T, U is right here. This structure is still the same. Okay. This is what's still the same, and that's what's making up this background. Are these bonds, these phosphates that are bound together all the way up through this, background. Now, again, these are two strands, separate strands, that are connected via hydrogen bonds. The hydrogen bonds are here. You can see them between the bases. This happens to be adenine, this happens to be thymine, and connecting them are these hydrogen bonds. And that's what's going on here. Now, some of them have more than, some of them have 3, which happens to be between G and C, and we'll see that on the next slide. And some of them have 2, T and A. And so that's also very important, because if you understand the composition of the DNA, you can understand how stable or unstable that it is. Of course, you can imagine something with 2 hydrogen bonds is going to be a little bit weaker than 3. And so when you take a gene, for example, and you sequence it and when you talk about gene sequencing, this is what it is these nucleotides, the order that they're in. Genes or DNA strands that have higher GC base pairing happen to be stronger than ones with TA. So you can relate that to other types of things like boiling point, melting point, etc. And so back to this, slide. Again, I didn't show you all this for no reason. I do want you all to know who these 2 individuals are, along with this individual and Rosalind Franklin. Photo 51, I want you to be familiar with as well. And the reason why it's on here is because it was taken by Rosalind Franklin. And at the time, women were not, common in science, so they weren't given respect. And at the time, James Watson and Francis Crick took credit for the things that she did, which was really the most pivotal point in discovering the structure of DNA, and she was given no credit. Now, of course, James Watson and Francis Crick were given the Nobel Prize for discovering the structure of DNA, and they don't give The Nobel Prize is after an individual dies. And she died shortly after the discovery, and the Nobel Prize was given probably in the 60s or 70s, and so she wasn't able to receive any credit. So, this person I certainly want you to know, and she will be tested on your exam. Linus maybe, James Watson, Francis Crick. You know, and Linus Pauling is interesting in itself. It's probably not the last time you'll hear him. You know, I thought I'd never hear him again outside of science. Turns out in my MBA classes, he had some theories on business. And so this name came up again, which was kind of interesting. And of course, in Oppenheimer, if you've watched that movie, he's a character in there as well. Okay. So nucleic acids, big picture coming back, you all understand that what we're talking about right now are macromolecules. We talked about carbohydrates, we talked about the lipids, now we're here at nucleic acids, which is what's making up DNA and RNA. I just showed you that picture of DNA and I said all those things, this is all it is here. And then you have those hydrogen bonds that are connecting them. But there's three components, and I did mention this yesterday, but this is more detailed. You have a phosphate group, and what you see here is, you know, you have a P and 4 oxygens. This one happens to have a hydrogen on it. And I've shown you all this before, but I really want you all to be able to identify this. This is the same thing as this. Now, this happens to be bonded to something, okay? So there's a hydrogen here. It's a little bit, I guess a little bit more neutral. However, free phosphates, similar to when we're talking about free hydrogens, this is how they're written. So whenever you're seeing this writing, understand that that's this here. This is a phosphate with 4 oxygen attached, and one of them is a double bond. Okay. And these phosphate groups are very reactive. And so, you know, I think this is the first time I'm introducing it to you all. I may have shown you the structure of ATP. But ATP, I brought it up to you all, is the energy source in the body. ATP has 3 of these things bonded together. And it's this group, because of its reactivity, that happens to release a tremendous amount of energy. In the structure of DNA, it making up the backbone, these phosphate groups attached laterally, so up and down, happens to make a fairly strong backbone to hold DNA. These oxygens bind tightly to phosphate groups. And so it makes it a good backbone. Then you have this pentose sugar, and this happens to be deoxyribose. Please remember that, because there's no oxygen here. In ribose, you would have an oxygen bound here. You would have 2 oxygens on each side. This happens to be deoxyribose, so you know that this is going to be part of DNA. Now, when I'm talking about RNA, it's the same structure, except we have an oxygen here on the ribose sugar, an OH group. And so there's 4 general types, and we'll see this on, so the pentose sugar, and then we have the nitrogen-containing base, which is what is the distinguishing difference in the sequences of DNA. So when we talk about one nucleic acid, this is it, but what separates one nucleic acid from the next is right here, and that's the nitrogen-containing base. There's 4 of them in DNA. Okay. There's adenine, guanine, thymine, and cytosine. Okay. And these nitrogen-containing bases bind right here, and each one looks a little different. Okay, so there's these 4 types of nitrogen-containing bases. This is what there's 2 major categories. There's one category called the pyrimidines, which happen to be a 1-ring structure. Okay, a 1-ring structure of the pyrimidines, and that's cytosine and that's thymine. And then you have the purines. which are two ring structures. There's guanine and then there's adenine. Now these are all nitrogen groups. The one important thing I want you to understand is that the purines bind, or the pyrimidines bind to purines. You have guanine that pairs with cytosine, and you saw it on the couple previous slides with the DNA strand. Guanine will bind to cytosine. Thymine, the pyrimidine, will bind to adenine. Now when I'm saying binding, what am I really saying? Hydrogen bonds, okay? It's not, we're not sharing electrons, but we're attracted to each other. The two strands are attracted to each other through hydrogen bonds. Then there's a fifth class of pyrimidines, so a single ring structure called uracil. And uracil is only found in RNA. So you can imagine, I don't have the figure here, and I don't think I have it on the next slide, but you can imagine uracil looks something like this, but the only difference is that there will always be a hydroxyl group or an oxygen bound to the pentose sugar. Okay? So I could draw a structure like this and put uracil here and say that this is uracil, what's wrong with the image? And you would have to identify, okay, it's not bound to a ribose sugar, it's bound to a deoxyribose sugar. Okay? So understand the difference. When you see uracil and you want to look at the nucleotide, you need to be able to recognize the correct nomenclature or the correct structure. There will be an oxygen present. Okay. And again, pyrimidines have one ring structure, purines have two ring structure. Guanine pairs to cytosines via 3 hydrogen bonds. Adenine pairs to thymines via 2 hydrogen bonds. That's very important to know. Okay? Because this explains the stability of DNA. And then, of course, I want to make a point too. Whenever DNA becomes RNA, what is that process called? DNA becomes RNA via transcription. So DNA becomes RNA via transcription. Did you drop this? Via transcription. What happens during transcription is the thymine is replaced to uracil. Okay? So thymine is excised from the DNA and uracil replaces the thymine. And so it would pair with adenine. So uracils now pair with adenine in RNA. And that's the first step. DNA transcribes to RNA, and then RNA translates to the protein. That's all DNA is doing. Going from DNA to RNA to protein, and those steps in between are transcription and translation. Okay. Now, I keep saying about the stability of these differences in hydrogen bonds. Sir, a very common question on your final and on my exams is if I had maybe 75% of guanine cytosine pairings, what would happen to the stability of the molecule? Or what would happen to the boiling point or the melting point? Okay? And the same with adenine and thymine. Understand that the higher concentration of guanine cytosine pairings you have, the DNA is more stable. Meaning it would take a higher temperature or more force to be able to rip apart those DNA strands, okay? Now, if you have less guanine cytosine pairings and higher adenine thymine pairings, that makes the DNA less stable, and therefore it can come apart easily. And when we get to the actual process of DNA replication, we'll talk about why this is so important, because there are certain portions of DNA that need to be pulled apart easier. Okay, to start the process. And so they would have higher thymine-adenine pairings, or higher concentration of adenine-thymine versus guanine cytosines. Okay, so this is important. And I put this note on my last class. It's just interesting to point out. I say that these things get a little bit more detailed. When I was towards the end of undergrad, so around 2022, they were discovering that there were a lot of modifications that were occurring on DNA after we had been born. So after the cells there were modifications that were occurring on certain cells. And so this is a cytosine, and these modifications, one was 5-methylcytosine, so on the 5th carbon, there was a methyl group put here. You all recognize that this would be a carbon, okay, with 3 hydrogen bonds. So they named it 5-methylcytosine, and then there was one called 5-hydroxymethylcytosine. So on top of the methyl group, there was added a hydroxyl group. And these were modifications that were occurring after DNA was there. And they were recognizing that these things were so common, but not only were they common, whenever the cell divided, they would maintain these 2 structures. And so now, it wasn't just cytosine, but then they identified these 2 things very recently as 2 new base pairs. And this happens to be a very heavy topic in cancer research because some of these modifications are what's making cells turn cancerous. Okay? And so I thought it was interesting. These are now considered the 5th and 6th base pairs. So there's not really 5 base pairs in DNA anymore, or 4 base pairs. There's actually 6. And then, of course, in RNA, there's still 4 base pairs, right? Because thymine is replaced with uracil, so there's still 4 base pairs. But these are, I don't think they're considered part of RNA. I think the groups are removed whenever it transcribes. That's just interesting. You don't need to know that for the exam. Okay, so that's DNA. It's nucleic acids, DNA, RNA. We're still talking about macromolecules. We're not really going into the breadth or the details of DNA. Okay, so don't think that we're doing it. We're just talking about the macromolecules. So the next one, of course, is proteins. Okay, and then there's that little excerpt there. Linus Pauling, for the longest time, had considered, and everybody taught in classes, that proteins were the genetic material. However, that was completely wrong. Although proteins are the most common thing in the body, they make up 10 to 30% of the cell's weight. So an individual cell is made up of like 30% protein. But an important thing I want you to understand is that when I say protein, a protein does not do one single thing. Okay? Proteins are doing millions of functions in the body. A protein is a way that you describe something. Okay? Just like when I say carbohydrates, you all understand that we have many different types of carbohydrates. Some of them aren't used for energy, right? Glucose is just one type. But there's other types of carbohydrates that are doing other things in the body that have nothing to do with food or have nothing to do with energy. It's the same thing here. Protein is a way to describe what the thing is. It's basically saying, okay, it came from DNA. But there are so many different functions. They have structural support. There's hormones that are proteins. There's receptors. There's some involved in transport mechanisms. And I could go on and on and on and on. The point is, when I say protein, you need to understand that it came from DNA. Protein is a way to describe something. It's not associated with a specific function. Okay. And they're, throughout the body, we've seen, without you all even knowing it, we've seen at least 20 examples of protein so far in this class. But importantly, proteins are made of this monomer called amino acids. Okay? So proteins is the macromolecule, and it's made of a monomer called amino acids. And so again, here are some of the many different functions associated with proteins, and this is not even an exhaustive list. Okay, there's some involved in structural support. In that case, it's collagen. There's enzymes that we've talked about where we're holding 2 molecules together so that we essentially are increasing the chances that they collide with one another that have a chemical reaction, and thus lowering the energy required for it. We have transport proteins. In this case, hemoglobin is one of them. Transports oxygen, which is present in blood cells. We have contractile proteins, and we'll talk about these in detail. This is involved in muscle contraction, communication proteins. We have proteins in the form of antibodies that are involved in the immune system. I mean, the proteins are on and on and on and on and on. The important thing to understand is they come from DNA. And this is what our DNA is making. This is what makes us us. Okay, so amino acids have a specific structure associated with them, similar to nucleotides. They have 3 main groups. The first one is an amine group. This is an NH2. NH2. The amine group is present here. Then we have an acid group. It's abbreviated like this, COOH, but here's the actual structure of it. And when I say acid group, what do you think is going on with this portion of the protein? When I say acid group, what do you think's going on there? There's no stupid answer. Somebody try something.
There's more hydrogen.
Okay, there's more hydrogen, but what do you think that this group is doing? That's describing the solution, really. But right there, what do y'all see that's unique about that? What do you think goes on here that makes that thing an acid group?
A double bond.
Okay, not necessarily the double bond, well, I'll get to that in a second. But there's something, we're missing something. There's a carbon, there's an oxygen, there's a hydrogen. What is making that thing an acid? What do you think happens here? Definition of an acid, like you said, is increase in hydrogen ions. So what is it about that structure that's making it an acid group? It has to be doing something, right? Everything has a purpose. It releases that hydrogen. Exactly. That's what's making it that acid group. So when I'm saying acid group, understand that this thing will not stay the same forever. It will release that hydrogen. And oftentimes, I know I have this written as an amine, but this is considered. So whenever you see this structure, and sometimes it's written as this, and hopefully now that's bringing in some thinking that this hydrogen is removed. This is called a carboxylic acid. And carboxylic acids and amines, there's some other ones. These are considered functional groups, and we'll talk about those later on. But you need to be able to recognize these two functional groups now, because these things have an acid group is just an abbreviation for carboxylic acid. Oftentimes people just don't want to write that; they just write acid group. But these two things have specific functions associated with them, and they're so common throughout all proteins that they are given names. They're called functional groups, and they have a specific function. Okay. So on all proteins, this is a common structure, similar to the phosphate and the ribose or the deoxyribose on DNA and RNA. This is the common structure. You have one central carbon, you have an acid group here, you have an amine group, NH2, you also have a hydrogen, and then you have this thing called an R group. And an R group, similar to the base pairs, the nitrogenous bases, is what's making each amino acid different. Now understand, these are the monomers. It's multiple monomers strung together that makes the macromolecule, which is the protein. Okay? So the R group is the variable group that makes the amino acid different. And there's many, many different types of amino acids. You all are taught 20 basic amino acids, and they only differ by that R group. Okay? Now, what do these R groups look like? They all look incredibly different, and they all have specific, characteristics about them. Some of them are polar. You know what that means. Some of them are non-polar. Some of them are charged. So you already know what type of bonds these might be forming. Okay? You already know the type of bonds that these already have, polar and non-polar. Okay, so these R groups look very different. And I'll tell you now, I don't want you to know the different R groups. However, your life will be a lot easier if you learn them today instead of later, because a lot of you will have to learn each one of these structures. Along, the name, the 3-letter code, the 1-letter code, and the structure associated with them. But I'm not making you all learn that, but you will see these again throughout this class, because a lot of them have specific functions. Because understand, why am I showing you all this? Because proteins are so abundant in the body, and each one of these R groups has a different chemical structure that allows it to interact differently with other molecules. This is the name of the game. These molecules are interacting differently with other things. They're binding to receptors differently. They're causing different chemical reactions. Okay? So, and again, you'll see these things because specific amino acids on proteins are very important for specific functions. Okay? Now, hopefully y'all can recognize that each one of these groups are the same. These functional, or these backbone groups. The ones that are highlighted are what's different. But you can see here, you have a carbon, you have a hydrogen, you have that carboxylic acid. Now in this case, the hydrogen is not there because it is releasing its hydrogen. And we'll talk about why molecules release their hydrogens and hold on to them based on different pHs. So depending on the pH, oftentimes an acid group will release its hydrogen, and then basic groups will actually gain hydrogen, and it's all depending on the pH. So that's why you see an NH3 here instead of an NH2 like I originally showed you all. Okay. Now, how do you go to these amino acids? This is based on a genetic code. So you all have 4 base pairs in DNA. Right? This is going back. Protein is in front of the process. All right, so again, do not forget this. DNA, RNA, protein. This is what's made of amino acids. RNA is made of a ribose. We can just put that there. Simply, you have U, A, G, and C. DNA is deoxyribose. And you have A, T, G, C. Okay. Now we keep talking about the code. The code is the DNA is like the blueprint, it's the genetic code. Well, these base pairs, a combination of them in sequence in DNA, is what's encoding what amino acid to make. And then that single amino acid will then, they'll combine together to make the full protein. Okay? So I don't, what I haven't shown you all though is that, and I don't know why it's not on these slides, is that 3 base pairs, so we're still at DNA, equals 1 codon. Okay, so 3 base pairs on DNA, A, T, C, those are the nucleotides, equals 1, what's called a codon. It's 3 nucleotides. 1 codon is equal to 1 amino acid. Makes 1 amino acid, which is what we're talking about now at the protein level. Okay, so 3 base pairs makes 1 codon. It's called 1 codon. That's what it's called, 3 base pairs. And 1 codon makes a single one of these. It encodes one of those. Now, how do you know what combination of base pairs on DNA make what amino acid is what I'm trying to get at. Okay? And so these here are the single amino acids. You remember I just showed you that they have a 3-letter code associated with them. And so these, valine, alanine, threonine, isoleucine, leucine, proline, these are the individual amino acids. Now, this was created a long time ago, and it allows us to figure out what codons make what amino acids. Okay, so you start here with the first letter, and say the first letter is C. Okay, let's say that the second letter is C. Okay, so we start here C, so we're in this row, and we figure out the second letter is C. And we'll say the third letter is C. So we look at this one. And now we're identified this row. CCC encodes for proline, 1 amino acid. Okay? That's 1 monomer of the protein. Okay? Again, protein is the macromolecule, that's the big picture. The individual brick is 1 amino acid. Okay? So, and you all know what C is, right? C cytosine. C cytosine, A is adenine, G guanine. You already know what these are, but it's the combination of those 3 that makes the protein. Okay. Now it's not a we'll talk about how we get there, but I'm trying to introduce the macromolecules, not the specific way to get there. But just so you understand, the nucleotides aren't directly making this. It encodes the amino acid. The amino acid we're getting from food. Okay, so we take in food and we break down the proteins into their individual amino acids, and those amino acids then go into each cell, and when we're making the protein, will line up with the specific codon that makes it. We'll talk about the process to get there, because it's important to understand. What I was about to say. But in second thing you all should be wondering is, why is there no T? There's only U. Okay. Well, we're not really talking about DNA at this point. We've moved on to RNA. We've replaced the T with U. And so what you'll see on here is the RNA code to become the protein. And so this is what our body does. We continuously are reading DNA to make a specific protein, which you all know the protein's the gene. Right? The gene is at the point of DNA, and the gene is making the protein, and it's reading it in sections of 3. And every single 3 codon, we're making the amino acid, and then the amino acids are bound together to make this protein. And what you all know about this protein is, with all these amino acids, it's an extravagant molecule that has all these groups with different characteristics associated with them going off the sides. Okay, you see all these large chains. And this causes all the amino acids to then interact with each other in a perfect way. This might interact with that one, and so it folds in a certain way because these interactions are being caused, oftentimes through hydrogen bonding and sometimes through covalent bonding. But that's what's making these proteins, and it's a specific sequence of these amino acids that make each protein different. Now, how do you make the amino acids bind together? There's 2 processes which you all already know. One is through dehydration synthesis. So you have 2 amino acids with those large R groups, and you remove water. That's why these groups are so important. You have an amine group, and then you have a carboxylic acid group. So you have a hydroxyl group and a water, or a hydrogen, that then leave as water, and they bind together. The carbon binds with the nitrogen. And what this is called is a peptide bond. You have to know that. These are the linkage points for the amino acids. And that's through dehydration synthesis. So, okay, I don't know what I was about to say, but they bind together through these bonds are called peptide bonds. So if I show you this structure, you should be able to identify they're bonded together by that's a peptide bond. And how do you get there? Through dehydration synthesis. Now, at the same point, to be able to break these amino acids down, we insert water, just like the hydrolysis reaction you'll have already learned. Insert water, and we break these amino acids apart into smaller components. Now, understand, again, that this process is constantly going on to make a protein until it stops. Cleaves it, and then we have a functional protein. Now, really, the adding of the amino acid is just the first step to making this large macromolecule. Okay, usually you start with 2 amino acids, which is called a dipeptide. I want you to know that. And sometimes proteins are just dipeptides. Some of them can be just dipeptides and circulate through your body, but usually they maintain the cells. And you have a tripeptide, which is 3 amino acids, and then polypeptide is 10 or more amino acids put together. And these are very common. And then a protein is considered 50 or more amino acids together. Okay. So this is similar to nomenclature of carbohydrates. You all remember we had monosaccharides, disaccharides, and we had polymers and whatever the carbohydrate, polymers, and then you got the macromolecules. And we talked about like glycogen, cellulose, starch, several. Or most proteins are between 100 and 10,000 amino acids. And 10,000, that's fairly large. But that's not it. So what I've been describing to you all is we're taking the amino acids and we're just stacking them on top of each other linearly. But I told you all that those R groups, those differences in the amino acids, will then interact with each other in a perfect way to make it fold a certain way. And when it starts to fold, they form different structures that we can identify. First one's called primary structure, then we move to secondary structure, tertiary, and then quaternary structure. Okay? And as you get higher and higher, each protein structure is bonded together by sort of different types of bonds. Okay? And so I don't want you to know these in detail. I really just want you all to get the general concept that we're going from a single amino acid chain. Okay, and here you don't see the extravagant R groups, you only see the backbone, but here are the R groups here. So you know that they're a little bit different. But they're bonded in primary structure. This is essentially what I've been describing to you all, the amino acids being bound together by those peptide bonds, and they're just linear. Okay, that's a primary structure of a protein. These are not typically functional proteins. This is just more trying to get the sequence of the protein. The second structure is called secondary structure. Okay? And these are made through hydrogen bonding. These are hydrogen bond interactions. So again, we're not talking about full bonds, we're talking about interactions that cause it to move in a certain way. They're attracted to one another. Okay, and they form two important structures that I want you to know. One of them is called an alpha helices, which you all have seen that in DNA. You've seen alpha helix, except in the case of DNA, it was two strands of alpha helices that formed what we call a double helix. Okay? When we're talking about proteins, this is a single strand. This that forms this alpha helix structure because of hydrogen bond interactions. Okay? These R groups on these amino acids start attracting to one another via hydrogen bonds, and they form this cyclical structure that we call alpha helices. Now understand this is still under the category of secondary structure. Then there's another type of structure called beta-pleated sheet. Okay? Now this all has to do with the R groups present on the protein. Okay? The differences in these two things have to do with the R groups. The different groups that are there that cause them to fold a certain way. These basically form sort of something, it's not shown that well on the slide, but they form something like this. Where again, we had this long string of protein, and for some reason, because of the gene, we have amino acids here present that want to interact with hydrogen bonds this way, instead of this way. Okay, in this case, the alpha helices, the way that the amino acids are placed, the hydrogen bond interactions are here. In a beta-pleated sheet, for whatever reason, and it has to do with the gene, the sequence of amino acids, the gene that's present, whatever protein it is, they have hydrogen bonds that interact here. Okay, that cause it to want to fold this way. And that's secondary structure. So those two things are under the category of secondary structure. Then we get to tertiary structure, where we have a mixture of alpha helices and beta-pleated sheets. Okay? And these can be functional proteins. Typically, secondary structure is not forming a functional protein. Okay? It's not very stable. And you remember we said that these molecules, just because of their interactions, they want to become in their most stable form. This is happening without anything occurring. This is happening just naturally. Almost like you put water and oil in water. These reactions occur naturally just because of the amino acids that are there. Now, a protein, you have to understand, one protein is a single strand. So there are some portions of the protein. So again, I kind of want to get this idea across to you, and you all have seen this image before when we were talking about synthesis reactions. Like the amino acids are like balls on a chain, okay? And it's this long chain. You all saw that they can get up to 10,000 of these. There are some stretches of amino acids that want to form hydrogen bonding this way. And so some portions of this string will form alpha helices. And then other portions of the string will form beta pleated sheets. And together, now, please understand that this is one single strand of amino acids. This is one gene. Okay? Now, on this ball on a chain, which is the best way to describe it, and again, you all know that you have these extravagant R groups that are extending off of every single one of these things. Some of them will fold in a way that they cause beta pleated sheets, and other sections of it will fold in such a way that they form alpha helices. And so that's what's giving us tertiary structure. It's these combination of these types of bonds together that's causing them to be this way. Okay, alpha helices and the beta pleated sheets together. Now, tertiary structure can be a functional protein. They can be a functional protein. Tertiary structure can. Now, tertiary structure forms what we call why am I losing? A globular or a globule, or sometimes, again, this is one string of protein. These are called globules, sometimes called globulins, or they're called subunits. So you could say globules or globular protein or subunits. Okay. Some people say globulins, and those would be more antibodies. Antibodies are called immunoglobulins. These are proteins that are essentially one string. And this is tertiary structure. Now, a lot of proteins have multiple subunits that come together to interact with each other. Okay, multiple subunits. Understand when I'm saying subunits, it's one string that can form these. Now, multiple of these can then interact with each other, and sometimes they're identical. Okay, sometimes they can be identical. Oftentimes they're not, but then they interact with each other, and they form a functional protein, quaternary structure. And this is typically what we're thinking when we say functional protein. It usually has multiple subunits. Or what you know now is that multiple genes can make a single protein. Because I'm telling you, a gene is one of those single strings, a single protein. Now at quaternary structure, this is multiple subunits together to form a functional protein. And we'll see many examples of these. Probably today's Tuesday. Tomorrow we'll certainly look into this. These are like oftentimes what our transporters are in the membrane, that are transporting things. But you can see that there's multiple subunits coming together. Okay, and oftentimes they're used as transporters in the membrane because they can form holes that can regulate things that are coming in and out because of the amino acids that are sticking this way. And so I want you to generally know what's going on here, okay? Primary structure, this is simply just peptide bonds that are on one string, like this ball on a string. Single amino acids that are connected by peptide bonds. Now when we start to get interactions via hydrogen bonds between multiple pieces, then we form these beta pleated sheets and alpha helices, and that's secondary structure. Now the whole string protein, where you have both beta pleated sheets and alpha helices on one subunit, is tertiary, and then multiple subunits that then interact with each other are forming quaternary structure. Okay, and this is oftentimes what we're calling a functional protein. Does that make sense to everybody? Okay, now again, I wish this thing would work. Okay, it's not going to work, but I think that this will be valuable for you all. So I'm going to take time to do it. And I might do this one more time in the class because it takes time. But again, this is, a good way to study. So what have we been talking about? What's the big picture topic that we've been talking about? Macromolecules. Now under macromolecules, what do we have? Carbohydrates. Now, carbohydrates, we have many different types. And, I might not, maybe we will talk about them as, you could determine them what? By their carbon chain, right? You had 5 carbons, 6 carbon, and these were the monomers. 5 carbons were called pentose, and that was what? Ribose, and you all might help me out, but what else? What? Ribose and deoxyribose. 6 carbons, these were called hexose, sugars, and carbohydrates can also be called sugars. You had what? Glucose, fructose, galactose, and then you had oh my gosh, disaccharides, maltose, and you all get the point. This is just, we're talking about one category, macromolecules. And under each thing, you can layer your understanding because they're all connected. I can even draw a line up here and talk about where are these things coming from, right? They're coming from our food. What system does that have to do with? Our digestive system. Moving through the tube, again, don't forget, always think big picture and let's work our way down because that's what we're doing in this class. Now I know it's difficult, you're coming to class every day and we're getting stuck in details, but never forget the big picture. And we can do that for everything. You can write everything you know that we've talked about in the last 3 days right here under this thing. You can talk about deoxyribose. Okay, we have a connection here. Whatever, and we have glucose that comes together to form maltose, and et cetera, et cetera. You can go on and on and on down the line. Now, what we've just been talking about is here. We have amino acids. You had 20 of them. Amino acids come from, I don't even know how I can do this. Think DNA goes to RNA via transcription. RNA made up of nucleic acids. You could have all kinds of stuff written over there. A, T, C, G, you can even write the pyrimidines, purines, etc. These nucleic acids, 3 of them equal a codon. 1 codon equals an amino acid. And those amino acids bind together via what? What type of reaction? No, not hydrogen bonds. Yeah, dehydration. Two dehydration synthesis equals dipeptide. And we can go on and on and on. Now, the very last step that we've been talking about is how do these things fold to make something functional, okay? Now, I can tell you now, I studied a lot like this. You should be able, because all these PowerPoints, essentially, I'm teaching them in this way. We're starting with big picture concept and we're working all the way down to the individual details, okay? So you all should, I know I don't remember everything. I'm not like, I have to work to understand this stuff as well. But the way that you all should be studying, again, is teaching yourself in this way. You can have a whole hour lecture just on this, or just on this. Okay, and you all should be doing the same thing every single day, on a whiteboard, on your piece of paper, and lining it out like a diagram, because we're going from big picture topic all the way down to the details. And the details of proteins were right here. This was the last step. Now, of course, there's a little bit more about how the proteins are made we'll talk about later on in the class. Don't lose sight of what we're doing. Okay. Anybody have any questions? Over. Okay. You all go take a 10-minute break, come back at 10:05. Somebody. Okay. So now we're jumping from sort of the chemical component to more of the cellular component, okay, which is what's making up the cells, or what's making up the body. Tissues are the individual cells. Okay, and so, it's important to talk about some of the ways that we got to this point of understanding what cells are, but I think you all can grow to appreciate by now is that when we're studying the body in biology, every single class is studying it at a different scale. Right? You have microbiology. We're studying at the microbe scale. Really focus on the cells and really focus on the cells that are causing disease. You have organic chemistry or chemistry, which is really focused on the chemical component of the body. You have anatomy, which is really focused on simply the structure of the body. And then there's physiology, focused on how these things are functioning together, which is more at the organ system level and the organ level. We go on and on. We have physics, which is also very much a part of what we got going on in our body. More at the chemical, maybe at the cellular level. But we study this at many different scales, okay? Now, we're about to look at it at the cellular level. And so, cells is really the foundational component of what we have going on in our body. And it's the functional unit of our body, much like when we talk about elements, the functional unit of those elements were atoms. At the chemical component of our cells, the functional unit are the elements. That's the functional unit of our cells. But in terms of our body, from what we can see, the functional unit are the cells. And so this individual, Robert Hooke, and I know it's hard to believe, but in 1665, so before we were even a country, he discovered cells, and he wrote a whole book on it, and it's pretty boring. However, so I guess we'll talk more about this in detail. But he made this microscope, and he was able to visualize individual cells in 1665, and then describe them. He's the first individual he might not have been the first individual to see cells, but he was certainly the first individual to describe them, and give it a name. However, what really are cells? Well, one is that they're the functional unit of life, they're the building blocks of what we have in our body. However, all cells come from pre-existing cells. That was also something that we'll talk about, the discovery of that, which was fairly important to how we understand what we know today. But organisms can either come as single-celled, and we know that. We see bacteria, there's a lot of protists that are like that too, which is very similar to bacteria, just a little different. And we have multicellular organisms, that's plants, animals, fungi, and you can't really go beyond that. But these have multiple cells associated with them. So single-celled organisms are organisms that can exist and function as one single cell. We are not that way, and neither are plants and neither are animals. Okay, so Robert Hooke was born in 1935, died in 1703, was an English scientist, and much like people at this time, they were really trying to figure out how life was occurring. So you had, I guess, that's why there's so many different people that had their hands in so many different things. Like we talked about, Aristotle was an incredible biologist and scientist, but he was also very famous as a philosopher. Same with Pythagorean theorem. He explained a lot of things that we know in biology, particularly in the reproductive system. And also known as a mathematician and a philosopher. So a lot of these individuals, the same was the case with Robert Hooke, were trying to understand what was going on with life, especially even before we were even a country, which is amazing to me. However, he created this microscope and took some cork off a tree, or this bark off a tree, they call it cork, and he looked under a microscope. And what he could see were these small, little, tiny things. Now, obviously, this is dead. Okay, so they were hollow. You couldn't see anything moving or anything functional in them. But he saw these tiny little building blocks. And he said that they reminded him of essentially cells in a prison. And so he gave it the name cells. Okay. And so this was the first, what we're going through is what's called the cell theory, which was established in the 1800s. But it took a lot of work from multiple different scientists to get to what we know now as the cell theory. And the first component was that cells are the basic structure and functional unit of living organisms, discovered by Robert Hooke and was described through this cork. Okay, that's the first one. The second, or really the third part of the theory is that all cells come from pre-existing cells. Okay, now I know that these things sound really elementary, but people spent their whole lives figuring out this one statement here. And so this individual, Rudolf Virchow, he might have been called Robert. For some reason I want to call him Robert Virchow. However, he had this idea. There was an individual who did an experiment in 1668. The idea was that cells could arise from nothing, or from non-living matter, or non-living material. And that's what people understood for a long time. Cells could arise from non-living material, which doesn't make any sense today, but that's what they believed at the time. And so this individual did an experiment, had an open container, put some meat in it, and wanted to see if the meat would produce more meat. Okay, so he had an open container, and when he had the open container, flies came and made maggots. In a closed cork-sealed container, the meat did nothing. There were no maggots that formed. And then when he put gauze on top, the maggots formed on top of the gauze. One, the meat didn't do anything. However, what it also showed is that the maggots were coming from the flies. Okay? It wasn't being arisen from the meat. So one, the meat wasn't reproducing, but also another point of it was that the maggots weren't coming from the meat. There wasn't a living organism that was arising from the meat, as which they had thought for so long, because you leave meat out and maggots would appear. What was described in this experiment was that maggots were coming from something alive, which was the flies, and they formed on the gauze. Now, Virchow got a hold of this and started to understand, okay, maybe these cells are coming, our functional units of life, which is after Robert Hooke, were coming from other cells or other living organisms. And so this is now what we describe as the third law of cell theory. Okay, and we'll get to the second one. It's not as important, it's later on. But this was the idea for so long, was spontaneous generation of living organisms from non-living matter. Okay. So those are two theories I need you to know, and I want you to know the people involved in those. Okay. Now, you can describe different organisms based on what we've already seen. You can describe them as single-celled organisms. Again, there's another class of single-celled organisms, and we have these multicellular organisms. This is what's called a phylogenetic tree. Okay, and so, in essence, we have multicellular organisms that have similar components to them. And so the idea was that they arose from something similar. Okay? And the same that goes for bacteria. Now, there's many theories that spin off of phylogenetic trees, and I'm not trying to go into that. However, what is obvious is that where you have organisms that reproduce so quickly, like bacteria, you have different variants of bacteria, and the same for viruses. Different variants based on genetic mutations that arise in real time. You know, we see, I know it's not on here, but you can, obviously, gram-positive bacteria can be like Staphylococcus, staph bacteria. You can have different variants that become antibiotic resistant. So that would extend off of this portion of the tree. So that's essentially what this is trying to show, is that you have multiple different types of organisms that arise from a single cell. They're similar in that they're single-celled organisms that then could have arisen based on different mutations that occur. And so you can describe them a certain way, and the idea is that there's some sort of common origin between all of these, but I'm just introducing the concept. So the first one is the prokaryotic cells. Prokaryotic cells are single-celled organisms. So I'm telling you right now that these cells exist as one, or these organisms, like us, the furthest line of organization in the body, the organism, is arising from one single cell. So you can imagine, if I'm drawing out the levels of organization for a prokaryote, what the things we would take out. What is one of the things that we would take out, the levels of organization? The organ system. Okay, it's a single cell. We take out the organs. It's a single cell. Okay, the tissues. Again, it's a single cell. So what would you keep? You keep the chemical component, you keep the cellular component to some extent, and then you keep the organism component. Okay, so you see what I'm saying? This is a living organism that's one single cell. Now, they have certain characteristics associated with them. One is that they have circular DNA. Very important. What is our DNA? It's a double helix. This is a single strand of circular DNA. In essence, if you took one strand of our DNA, made it a lot smaller, and made it a circle, that's what a prokaryote has, particularly bacteria. The other thing is that they may or may not have flagellum. And what a flagellum is it's a protein that extends off of essentially the butt of a prokaryote, and it contracts, not muscular contraction, but the proteins will then flap around like a propeller. And that's how these things become motile or mobile. So some may have flagellum that allows them to become mobile. The other thing is that they have no membrane-bound organelles. And hopefully you all can understand what that means when we jump to the cell, and I've introduced the concept of the cell already to you all, but you see all of these, now these aren't organelles essentially, but you have all these things, the nucleus, the mitochondria, the endoplasmic reticulum, and I told you that they all have membranes associated with them. The plasma membrane wasn't just around the cell. These things, like mitochondria, endoplasmic reticulum, nucleus, they're called organelles, but they're also membrane bound. They're encapsulated in a membrane, all of these things. So you can imagine those phospholipids are all around here as well, that membrane bilayer. Prokaryotes do not have that. They don't have any membrane-bound organelles. So the first thing you should know, their DNA is just floating within their cytoplasm. There's no nucleus encapsulated in these, and we'll look into that a little bit. They also have ribosomes. Now, sometimes people get the idea that ribosomes are organelles. They're not. These are what's making the proteins from DNA, and we'll talk about that in detail, but they have ribosomes, and they're generally the smallest, simplest cells. Incredibly simple, and so they're believed to be like the earliest organisms on the planet. And bacteria and archaea are considered prokaryotes. You remember we talked about yesterday, there was something called electron microscopy, where you could see like heavy details on cells. What we were looking through were light microscope. This is an electron microscopy that happens to be given color. They're black and white for the most part, but you can add color to them, and you can see extreme details. Now, not super detailed to the chemical level. The impressions that you're seeing, one is the phospholipid membrane. You can kind of make out a little bit of phospholipids, but the majority of this roughness is the proteins that are extending off the membrane and whatnot. And we'll see a little bit what we're talking about. And when I say protein, you should know that some of those things are 10,000 amino acids. So you can imagine that this is certainly not showing you any bit of the chemical component of a cell. Okay, then you have eukaryotic cells. These are organisms. So again, organisms that have multiple cells associated, not just one. They can't exist as one cell. Plants, animals, protozoans, fungi. There are some fungi now that are single cell that can actually be fairly large, that they've discovered. But we have a cell membrane. With membrane bound, we have an enclosed nucleus, which is important because you saw in bacteria the DNA was floating within the cytoplasm. And we have membrane bound organelles. Okay. within our body, humans, we have, it's not just as simple as having cells, okay? We really have two classes of cells. We have sex cells or germ cells. These are sperm and eggs, which are what's allowing us to propagate. And then we have somatic cells, which are essentially everything else, okay? Sex cells can also be called germ cells, and somatic cells, there's not any other name for them. And typically, whenever we're talking about cells, we're talking about somatic cells, unless I'm specifically emphasizing sex cells, and that goes for almost anybody. So, you begin life as a single cell, right? The egg. The egg, of course, is a sex cell. This is a germ cell that then becomes penetrated by one sperm cell. Now, there's thousands of you that come and attack this egg and try to get inside this cytoplasm. Okay? And what I have written here, and this is just for y'all's information, is you have about 3 layers surrounding this egg. And one of them is called the zona pellucida. And once an egg, or once a sperm, which also has that flagellum, depending on which one, and nobody understands why one gets chosen over the other in terms of going into the egg, like what's going on at the membrane that allows one to go through. Some say it has to do with motility of a single sperm cell versus the others. But it penetrates the zona pellucida, and the zona pellucida is this sort of crystal-like protein that surrounds the egg. And for whatever reason, once one sperm cell penetrates that zona pellucida, the zona pellucida, which is a protein, so you know it has an extravagant amino acid structure surrounding this thing. For some reason, that protein, like a crystal-like structure, turns very rigid all of a sudden. And then every single sperm cell that's around, almost like, I don't even know how you would explain it really, almost like a wall that becomes, maybe all of the walls down, and then all of a sudden once somebody gets in, the walls go up. The structure of the protein completely changes around this entire egg. That all these sperm cells that are trying to penetrate the egg then cannot get in anymore once one enters. Now, once that happens, this single cell divides into 2. Okay, and then these individual cells then divide into 2 others. So now you got 4. Okay, and this is how we're becoming a human being. Now, the question begs itself. One is what you all should know already, because I've told you all multiple times now, is that every single cell has every single bit of your DNA. This cell has an identical DNA to that cell. And I think I brought up this idea to you all, is how does the cell then become skin cells? How does it become kidney cells? How does it become cardiac cells? How does it become digestive cells? What is that that's making the difference? If everything has identical DNA, just like I have an identical DNA to a twin, we're going to look very, very similar. But what is it that causes these cells to form fingers, to form heart cells, to form digestive cells? And that's not necessarily a question anymore. And that's where we get into what's called cellular differentiation. So we have these single cells, and maybe I should. That process of the cells becoming different cells is called cellular differentiation. But I do want to introduce this, because this is a figure that explains it a little bit better, is that you have those 2 cells. Once the sperm penetrates the egg, it's called a zygote. It's a 2-cell organism. And then those cells divide into multiple. And it divides in such a way that you start separating layers of cells. And it becomes essentially a hollow cavity also. The cells form a wall, almost like a sphere. And then you have this hollow cavity inside that actually forms like a yolk type of thing. I know it's kind of weird. But then these cells become what's called pluripotent stem cells. And pluripotent means that it can give rise to any type of cell. These are individual cells that have the potential to become any type of cell. Okay? So when I say pluripotent stem cell, this won't be the first time you see that. This has the potential to become any type of category of cells. And they do split off into further categories, like into, nervous system tissue, and then those cells have a specific name that can only become like brain cells or spinal cord cells, etc., associated with the nervous system and the age. However, when I'm telling you pluripotent stem cells, they can give rise to any type of cell. And they're very, especially in adulthood, they're very uncommon but not unheard of. But this is what's going on as essentially what's called a blastocyst, and there's multiple phases that's going on here. But a blastocyst, I think, is like 32 cells or 64 cells, and these start to form pluripotent stem cells, okay? And then these pluripotent stem cells will become 3 types of germ tissue. Okay, so these are tissues, but these are early on. One of them is called ectoderm, one of them is called mesoderm, one of them is called endoderm, and I do want you to be familiar with the names of these, not specifically what they're giving rise to, okay? Now what's interesting is that these do have identical DNA. So what's causing them to differentiate? Well, different cells, for whatever reason, and we'll get into a little bit of what's going on specifically at the DNA to allow it to become that way. But what's going on is these DNA are secreting, they're creating a protein that then these cells are releasing into this yolk specific proteins. And you don't have to know the names of these. Like I'm telling you right now, these individual proteins, people have spent their entire lives on describing this one track going here in even further detail. So this is not important to know, but I'm trying to introduce the concept, is that these cells start releasing specific proteins, again, made amino acids, that then, based on the concentration anywhere in this blastocyst, so anywhere within these 64 cells, maybe some of this OCT3/4 is present here and affecting these cells, binding to those cells. Maybe some of this SOX2 is over on this side. And they're and listen, I'm being very general, but this is explained very detailed. They cause these cells to become these 3 germ layers depending on where they are. So some of these cells start secreting some of this, maybe some of over here, and cause these cells to become that. Okay? Some of these cells may start secreting this and cause these cells to become this, the ectoderm cells. And then as they become mesoderm cells, they start secreting their own stuff. Activin A, BMP4, FGS, which we'll learn about, Wnt. And then they start going down into further differentiating into their specific cell types. And I hope that this concept makes sense to you all. That these cells start secreting, it starts off with a small number of proteins that they start secreting, and they start diffusing differently in this blastocyst and cause these cells to move down these different paths. And then they start secreting more and more stuff that then cause them to move into these different paths. And it's the combination of these proteins together that then make them specific to whatever they are. Now, specifically what they're doing is essentially some of these proteins are involved with going to DNA and turning off certain genes. Like for example, on a cardiac cell, you don't necessarily need, we'll say, you wouldn't necessarily need like melanin or something that you need on the surface of your skin to protect from UV rays, right? So some of these may turn off those genes and turn off certain genes so you don't make proteins that would make you into a different type of cell, okay? And this is incredibly detailed. This is years and years of classes to understand each one of these, and it's really interesting if you're interested in that type of stuff. But the concept is you have those cells and they become each one of these types of cells, and they look incredibly different. You all have seen cardiac muscle cells. They look incredibly different from liver cells or from skin cells or bone cells, okay? And so it's important to grow an appreciation for this. But you all have seen that they go through steps of what's called differentiation. They just don't become cardiac cells. They go through these various steps. And so what causes them to become this, big picture, is that they express specific genes. They also turn off a lot of genes that they don't need. Okay? And the process of having a gene expressed is called genotype. And you all need to know this term, is that this is your cell's genes that have been turned on. And this is a statement I've been giving to you all throughout this lecture. And then there's this idea of gene penetrance, which describes what a phenotype is. So I've talked about a genotype. A genotype is the specific genes in the cell that are being expressed. Now, not all the genes are expressed in a cell. Different cells have different genotypes. Again, like I've already told you, cardiac cells have a different genotype than the muscle cells. They're expressing different genes. Okay? Or cardiac cells have a different genotype than skin cells. They're expressing different genes. They're turning off different ones and turning on different ones. Now, genotype, when a genotype is expressed, it becomes what's called a phenotype, what it becomes. Okay? So, phenotype is really describing, instead of the gene would be, I say, insulin. Insulin's a gene. The gene would be insulin. Okay? Now, the effect of insulin, how much insulin you're making, what is insulin binding to, whatever the gene is doing is called the phenotype. And sometimes the way to understand it is G is gene, P is, you could say, physical. It's the physical being of the protein. What is it doing? Okay? How can you describe it? And so, oftentimes, especially if you've taken biology in high school, they describe, like, the easiest thing is to describe coat color on dogs. You have the gene for, maybe a white coat color, and then the phenotype that's expressed would be the white coat color. Okay, the gene would be the specific gene that's making the white color. Okay, and so I don't want you to think too much on that, but maybe do know the difference between genotype and gene. Gene is the gene, phenotype is the physical expression. Now, okay, we'll talk about this plasma membrane because I want you to leave you all this. But you all have seen this figure multiple times. You've seen a cell. We'll talk about each one of these in detail, but for the next slide, I really want to focus on the membrane components of the cell. Now, for exam, the exam coming up, you will have to know the functions of each one of these things. So it might be a good idea to go ahead and get started on the functions of each one. And I'm talking about really just general functions. But certainly we'll talk about that in detail tomorrow. But I really want to focus on the cell membrane. And so we're focused really right here. What's surrounding the cell. And so the way that I've introduced it to you all is that we have those phospholipids. Now, phospholipids is really just one component. Okay? You all have one in the second component, and that's cholesterol, and hopefully you all can see those 4-ring structures embedded here in this membrane. Remember, the component of steroids that you have to know is that it's a 4-ring structure. Okay? This is the definition of what a steroid is that base structure there. Now the difference happens to be here. Okay, so we have the plasma membrane, but there's a lot of things going on in the plasma membrane that are very important to understand. And so we have proteins, so we talk about phospholipids and cholesterol, but we have these proteins that are in the membrane. Okay, and these proteins are doing a lot of different things. And what I want you to know is that there's 3 main categories. We have integral proteins, which are proteins that are in the membrane. Okay? Integral proteins is describing a protein that is present in the membrane. And when I'm saying protein, remember we're talking about amino acids. And amino acids are coming from DNA. So we have all these proteins that are in the membrane. We have integral proteins, which are all these proteins that are in the membrane. Now, there are 2 ways to describe integral proteins. You have transmembrane proteins that span the plasma membrane. In essence, it's facing both sides of the plasma membrane. transmembrane. And then you have something called peripheral proteins, which are present only on one side of the membrane. Okay? They're not embedded into the membrane. They're present only on one side. Now, these things are all doing specific functions, and we'll talk about this. Happens to be a transporter here. And understand, you all see this is a large globular thing. But I told you oftentimes these proteins that are in the membrane have multiple subunits associated with them. So you can imagine right now how an artist's depiction can make you lose sight of what's actually going on with these proteins. Everybody see that? Don't forget that we have structure. This is a quaternary structure protein that has multiple subunits associated with it. Even here, that the artist isn't even depicting to you. And you can tell how much chemicals are lost in this thing. Now, that's peripheral proteins, and we have transmembrane proteins. Also, we have a tremendous amount of carbohydrates on the surface of the plasma membrane. A huge amount. And we know already what carbohydrates are. Okay? There are tons of them, and they become, now this isn't glycogen. I know I showed you glycogen looks something similar. Glycogen is much bigger. And this is what's being stored inside the cells. But we have glycolipids and we have glycoproteins that are present on the surface of the cell. And they're so present that it actually forms this layer that goes beyond the plasma membrane. Okay, and this was recently discovered, and it's called the glycocalyx. So this plasma membrane has carbohydrates that are extending so much off of the phospholipids that we now have a name for this layer. So now you can see carbohydrates aren't just for energy. This is not to give the cell energy. And what the glycocalyx is doing is allowing for us to identify cells. Now, there's a huge amount of glycoproteins and glycolipids present here. And when I'm saying glycolipids, these are lipids that have carbohydrates associated with them. These are proteins that have carbohydrates on them. Okay? Glycoproteins, glycolipids, and we'll talk about each one of these in detail. But they form this layer called the glycocalyx. And the glycocalyx is a portion of the cell that's identifying it as me. It's identifying your cell to a number of proteins that are moving through our bloodstream. Okay? It's essentially an identifier for cells. So these things get very complex. And some of the most common glycoproteins that you all know is, one is very common, hyaluronic acid is a very common. It's used in skincare. These things are present on the membrane. Hyaluronic acid is a real thing that's existing within the cell at the chemical component. And what it happens to do is just bind a lot of water and help keep this layer on top hydrated in the extracellular environment. So it's keeping the extracellular environment between the cells hydrated because it binds a tremendous amount of water. But okay, we will. There's an extra. I would recommend to you all, looking through these, because we will move very quickly through each one of these organelles within the cell. Look through each one, look them up in your textbook or look them up online, and just give them a small function associated with it. Okay. I will see you all tomorrow. I'll try to get a study guide out tonight, which I will get one out at some point today. But understand, do not keep a study guide from keeping you from studying. The study guide is just to confirm that you've been learning the right things. All right, but you all have all the information you need for an exam. Oh, sticky notes.